Hi there, let's deal with topic 5 in our book for diversity. In this topic, I'm going to explain DNA and its role in helping us pass on traits, as well as review mutations and talk about some genetic technologies like cloning, genetic engineering, as well as genetically modified foods. Let's review for a minute. We've talked about how traits might be passed on, and sometimes that might be beneficial. However, it also might be lethal in the case of the African population and sickle cell anemia. Now, sickle cell anemia has to do with the following. If you have the disease, then your red blood cells kind of squash in like this in the shape of a sickle. And there are three forms. You could be sickle cell, which means you're recessive for that gene. So your blood would have two S's. If that's the case, you're going to be really sick and possibly die because your blood can't carry oxygen, which means you're anemic. You could be intermediate, which means you're a carrier, or you could be normal. Now the problem with this is if you're normal, which means you don't have the sickle cell, then you're susceptible to malaria. In Africa, it's a disease passed on by mosquitoes. So these people have a greater chance to get malaria, which could cause them to be sick. And these people could get sick through sickle cell anemia. So the most well-adapted people would be the carriers of it because some of their blood, but not all of it, would be sickle cell. So in essence, African descent people only have a 50% chance of having a normal intermediate, which would have a better lifestyle than someone who was no sickle cell, or unfortunately someone who had most of their blood in sickle cells. So just as a review, some traits have more than one possible allele. We've been talking about how we can use Punnett squares to help predict outcomes in regards to offspring. Remember, this is just the probability. That doesn't mean that necessarily when pea plants produce 4 offspring that they absolutely would have, for example, a 25% chance of a short pea plant. So it's just a probability. As I've talked about before, we have a 50-50 chance when a married couple has a child that it will be male or female. There's a 50-50 chance. However, there's lots of examples of families who have 5 boys or 5 girls. And so, again, what really happens isn't necessarily totally based on the Punnett square. Some organisms' characteristics are due to several gene locations, for example, the dog's fur. So not all traits are cut and dry. In other words, they don't have a discrete or a one or the other kind of format. Let's get into topic 5 now, and first thing we want to talk about is DNA. And we need to know the structure of a DNA molecule. So let's take a step back. Here's a cell, and inside the cell of eukaryotes, which are organisms that have nucleuses in their cells like us, is genetic information. So inside there our chromosomes. And what is a chromosome? It's wound up, coiled DNA. And so when we blow up that DNA molecule, on the DNA are certain sections called genes which help govern traits. And a gene has, in many cases, two alleles. So this might be a gene on the strand of DNA on the chromosome. So this might be right there. And that gene might govern a trait like your earlobes. So DNA, which carries instructions that allow cells to make proteins, is made up of 4 chemicals. Okay? And these chemicals, or the chemical bases, help govern what proteins are made for our bodies. So let's get into these chemicals. DNA is sometimes thought of as a twisted ladder, and the rungs on the ladder represent the base pairs which help determine the genes because they make proteins that do that. There's a pattern between the base pairs that you need to be aware of. So the 4 base pairs are A for adenine, T for thymine, C for cytosine, and G for guanine. But on the DNA ladder, A adenine and T thymine can only join together, and C cytosine and G guanine can only join together. So if you look, you will only see C's and G's and A's and T's. However, each individual, the orders of A's, T's, or T's, A's, and C's, G's, and G's, A's might be different depending on your DNA and genetic makeup, which then helps determine a gene which makes our traits. Here it is again. So Watson and Crick were the people who did lots of studies and investigated the model of a DNA. So we have the sugar phosphate backbone. So phosphate, sugar. And then we have these nucleotide base pairs, C's and G's, A's and T's. Let's look at a quick video clip on this from YouTube.
DNA contains 2 strands of building blocks called nucleotides, arranged like a spiral staircase. Each nucleotide includes 3 parts: a phosphate group, a sugar molecule, and one of 4 bases: adenine, guanine, cytosine, or thymine. The sugar phosphate bonds form the double backbone of the molecule, the handrails of the staircase. But we find the genetic key to DNA in the steps of this stairway, the nitrogen-containing bases. These bases link up using hydrogen bonds in a very specific way. Adenine will bond only with thymine, A to T. Cytosine only bonds with guanine, C to G. While these basic pairings never change, the order of the pairs along each strand varies greatly from one species to the next. In this elegant design, we see how nature stores the instructions to build all living things.
Let's go back now to our PowerPoint. So I want to talk about the difference between mitosis, which is how our cells grow and divide, and how it's similar to binary fission, which is what we looked at earlier on for sexual reproduction, or no, asexual reproduction. So mitosis is very similar to how bacteria and some simple organisms carry out their reproduction. So here are the chromosomes in binary fission. Notice they're not in a nucleus because they're prokaryotes. And eventually, the wound up chromosomes copy each other. Cha-ching, they double. So you create an exact copy, and then the cell wall, or the cell membrane, pinches in, and you create two identical cells. Well, that's how mitosis works in our bodies to help grow what we call our somatic cells, which are cells for growth and repair. So they've simplified it, but here we have just two strands of DNA. Step one, they replicate, just like it does right here. And then they line up along the equator of the cell, and the cell pinches in, and you make two exact copies of the cell. Now, if it was in our gametes, which is egg or sperm, what would happen is our gametes, remember, have half the number of chromosomes. So instead of 46, they have 23. So that would be, in our gametes, we have, instead of mitosis, we have a process called meiosis. And in meiosis, we divide again. So these two cells here would divide to create two new cells with just one chromosome. And this would break off to produce two new cells with just one chromosome. So the difference between meiosis and mitosis is as follows. Meiosis is replication and single division to make two identical cells, and it's for growth and repair. This occurs in our body or somatic cells. Meiosis only occurs for our sex cells to create gametes, egg or sperm, and it has half the number of chromosomes. And it's double division, so it would divide a second time. Here it is again. So here's a picture of meiosis. So the creation of your gametes. So DNA replication, and then single division, and no replication again, but division again. So through meiosis, we create our gametes, which in humans has only 23 chromosomes. They fuse to make a zygote through fertilization. Through mitosis, this cell divides, eventually forming a baby with 46 chromosomes. And through mitosis, the baby grows up, gets bigger, creates an adult. Let's review nurture versus nature again. These are two twins. There's so many similarities between the twins, and we would call that nature. However, how you grow up and what your environment is like also determines who you are, and we call that nurture. So if this twin grew up in a different family than this twin, and they were raised in different environments, although they would have very similar traits, there would be lots of things that would be different about them. For example, this twin grew up in a very sunny country. This twin grew up in the United Kingdom. So notice this twin has freckles and her face is a lot more wrinkled because she grew up in Brazil, whereas this twin grew up in the UK where there's less sun. So this twin has slightly different color hair, not due to nurture, where she was brought up and nurtured. With this twin is different. So again, nurture versus nature. Traits that are not influenced by genes, that's nurture. Their likes and dislikes of clothing and music, their diet, how smart you become, your swimming abilities. There are traits, though, that are influenced by genes, your eye color, for the most part your hair color, and your physical characteristics. Sometimes, though, there can be mutations on genes which would cause variation as well. So mutations is an important part of genetic variation. Half the genes in our body come from our mom, half from our dad. This form of mutation is an error that arises when genetic information is copied. You can develop mutations throughout life too. X-rays might cause mutations, and then they can be passed on to your kids. So let's get into genetic technologies. One type of genetic technology is